Hey, what is going on? It's me, and it is time for the final video of the month, February 28th, 2022. Just for a little non-bio context, well, not really bio, the war in Ukraine is still going on. I feel like I'm like a Cold War commentator or something from 30 years ago, 35 years ago, talking about how the Cold War is still raging, etc., etc. But I guess some people could never really get over the Cold War, right? So, anyway, without going too deep into it, yes, there's instability in the world, but that doesn't mean life won't go on, even if there may be some questions about the state of life now. We still need to talk about all the components of life. So I am actually doing one more diversion from the cell cycle. I do plan to go back to other pieces of the cell cycle later, like I left off at anaphase, or the beginnings of anaphase, and essentially how anaphase is progressed through the activation of the anaphase promoting complex, which does a bunch of stuff to physically cause chromosomes to separate, and which causes I haven't gone too deep into it, but it essentially causes the proteins that bind the chromosomes together, called cohesins, to separate through activating something called separase, which does the separation by dissolving securin. Okay, so basically the anaphase promoting complex dissolves securin, destroys securin. Securin was securing separase. Separase then goes and separates the two chromosomes, or the two sister chromatids, from a chromosome by breaking cohesin, which kind of wraps the chromatids together. And by doing that, there's another protein called condensin involved, which I'll get to in a second. But the two sister chromatids come apart because the mitotic spindle is attached to them, and the mitotic spindle was, to the best of the cell's ability, verified as being able to pull the two chromatids apart because the presence of anaphase promoting complex says so. And the two sister chromatids go to the centromeres on the opposite sides or opposite poles of the cells via the polar microtubules that are pulling them. Okay, so it's like a kind of a ribbon cutting, almost, in a way. I think that's one way of thinking about it. But we should also just briefly, no, that's what I wanted to get to, and I guess I kind of did. I did want to mention condensin also. Condensin is another molecule of interest, which, as the sister chromatids are getting pulled apart, the condensin molecules, and there's different kinds of condensin, but they, with the help of attached motor proteins, prevent the sister chromatids from flying apart, and the momentum from destroying them, I guess, or warping them as they fly to the ends of the chromosomes. So ends of the or they fly to the centromeres, or the centromeric poles of the cell. So there's so many things going on, but yeah, there has to be a way of keeping those together. So those are the major top level things involved until telophase occurs. And I really haven't gone into telophase at all, but at that point, telophase is where the two cells start distinguishing themselves, and a nuclear envelope forms around the chromatin. The chromatin disassociates, the cell cleaves along a cleavage furrow, or cuts in half, and then the two cells finally separate from each other via cytokinesis, which is like the very end part of telophase. So, and then therefore the two cells have new cell life cycles since cell division is complete. So that's basically, and I'll go more into all those pieces later on. But I'm jumping a little higher level now because I think it'll help understand things later, into going into the cell structures a little bit. And today, like, it's actually, this is going kind of back to bio 1 type stuff, but we're talking about the nucleus. And the nucleus obviously is where everything happens, right? All the major functions of the cell occur, right? And that's where proteins get synthesized. And this is a eukaryotic function, right? Eukaryotic cells really have a nucleus, but the nucleus has a bunch of pieces to it, including a bilayer nuclear envelope, which the nuclear envelope itself has quite a number of complexities involved. A lot of them are in the form of the channels that allow entry of different substances into and exit out of the nucleus. There's its own complexity there. There's also lamin protein, which is attached to the inner nucleus, which is involved or indicated in the development of progeria. So there's many things that lamin does. So a lot of it has to do with interaction with the chromatin, which is, chromatin is all the DNA, right? That's within the nucleus, and in this diagram, it's these purple strands floating around the inner nuclear space, and then there's a nucleolus, where another very important organelle where the RNA synthesis actually occurs. So these are controlled environments, sort of, that are packaged within other controlled environments. So it essentially, it's a clean room in your electronics products factory, right, where the actual synthesis of the molecules occur, right? It's the synthesis of the blueprints, maybe, or whatever. There needs to be a specific environment. But then after these major components of the nucleus, then we go out a little bit and we see the endoplasmic reticulum. So again, this is a high school biology revisit, but we know about the rough ER and the smooth ER, which are two different, very important organelles which surround the nucleus. And I guess they're called reticulums because they're kind of like curvy passages or chambers. I don't know, but just as a reminder, the passage space is called the lumen, the inner space, and the inner lumen of the endoplasmic reticulums, specifically the rough ER, is where the proteins that are created by ribosomes are folded. So the shape of these channels help with folding of the proteins. But you have to remember the rough ER is called the rough ER because there's ribosomes peppered outside of the rough ER, and as the ribosomes create proteins, they translate it into the space within the rough ER, the lumen space. So another thing that's interesting that we got to think about is that how much of this is going at the same time. Like, there's not one ribosome manufacturing proteins, there's several of them. And essentially, how does the endoplasmic reticulum keep the proteins straight and separated from each other? Because these proteins probably have charges, they probably have interactions, van der Waals forces, other things that can happen between proteins. So there's probably a fair amount of complexity in this assembly line process here that goes on in the endoplasmic reticulum. So they don't go into that in basic bio, but at some point we'll go into that. But besides the point, so these proteins are in the rough endoplasmic reticulum. Eventually these are they go through modifications called glycosylations, and glycosylations are basically the adding of a sugar of a different variety to these proteins. Okay, so sugars are added to proteins in order to activate them, and that's like a whole separate lecture in itself, and that's also very protein specific. But a lot of proteins achieve some sort of active capacity, like the ability to do certain motor functions or whatever, through the adding of these sugars. this is glycosylation, has multiple forms, but it essentially occurs within the endoplasmic reticulum. And just a little bit more on glycosylation because it's interesting on how this occurs, and where, because proteins are composed of amino acids, right? And there's specific amino acids, which acid sites, which can be glycosylated on a protein. Okay, so basically glycosylation occurs, turns a protein into a glycoprotein, or basically a protein with little sugar attachments, or a sugar attachment, and then the protein does its thing. The protein has 3 amino acid sites where glycosylation can occur. And so, like, remember, there's several different amino acids. So we have threonine and asparagine, and serine are basically, the serine is not pictured here, but basically glycoproteins are linked via other proteins to a specific polypeptide chain through which would have like a threonine side chain or an asparagine side chain. So this guy is linked to this guy is linked to this guy, and the location of linkage is important. Important distinguisher of, the function and like what kind of linkage can happen. So threonine can get linked to a sugar via its hydroxyl group here, right? One of the alcohols or hydroxyl groups attached to the sugar. It's bound to the oxygen of the amino acid, the carboxylic oxygen, and that's called, what is that called again? That is called O-glycosidic bond, O being oxygen. Okay, so in the case of asparagine, there's a different type of bonding called an N-glycosidic bond, which occurs. And in that case, the sugar gets linked to the nitrogen, terminal nitrogen of the asparagine molecule. The carboxylic oxygen, the carboxyl group is left alone, and nitrogen is covalently linked to what's called the anomeric carbon of the sugar. So we don't need to go into all the organic chemistry nomenclature right now, but just to let you know, there are these 2 types of linkages that occur, and these occur in the endoplasmic reticulum, and eventually they start there, and the endoplasmic reticulum, if you remember, and we'll go into it later, packages the proteins to get sent over to the Golgi apparatus or Golgi complex, which has more of these glycoprotein events, glycoproteotic events occurring, which then finally allow the protein to be fully activated via different sugars, etc., and then packaged and get sent to their final destination. So we'll get into that in another video. This video is 13 minutes long, but yeah, thanks for listening. There's a lot to talk about, and we'll go down this route for a while before we get into the actual DNA synthesis parts, because those are really the most important thing, I think, that we need to talk about. Okay, I'll see you around.